Now you can do the electrophoresis. Okay, so we make all these copies of this target sequence that's relatively short. Okay, the target sequence is going to be less than 20,000 base pairs, which I know doesn't sound like a lot, but remember, you're talking about DNA. Remember, your cells, which you can't even see without a microscope, have 6 feet of DNA in them. 20,000 base pairs is a drop in the bucket. Okay, so the target is going to be between 2 primers. Okay, and between these 2 primers, we're going to copy this target. Now, you cannot use PCR to copy the entire chromosome. Okay, and to do this process, so you're going to have, you see up here in your starting materials, okay, you're going to have DNA polymerase, you're going to have the 2 primers, you're going to need some nucleotides, okay, and you're going to have the sample of DNA. Let's talk about why we would actually need these. Okay, well, DNA polymerase is going to do the pairing up, right? Okay, so we're going to need the DNA polymerase here to add the nucleotides. The primers, remember, they start the replication process, so I need some primers so I can actually copy it. Okay, and then I need some nucleotides. I need some raw materials. I need to be actually be able to base pair things together so I can get the new strands of DNA. So you can see in the picture, it walks through the steps here. So you have this targeted sequence, okay, and there's the strand itself will be heated briefly, and that'll break the hydrogen bonds and separate. It'll act like the helicase. It'll break the hydrogen bonds and separate the DNA strands. Okay, and now we can cool it down so the primers can actually hydrogen bond. And the primers can start the replication process, use the DNA polymerase and the nucleotides to complete the strands of DNA, and just keep going through that process. And as we keep going through that process, we're going to be doubling our numbers of DNA every single time we go through this process. So you went from 1 to 2 to 4 to 8 to 16, okay? And like I said, you exponentially are growing your products here for this. So these PCR products, they can then go into the electrophoresis machine, and they can be analyzed for what are called STRs. Okay, and STRs are short tandem repeats. Okay, and so what this is each individual has a unique pattern of these STRs. And so for a single set of primers, they're going to have 2 PCR products if they inherited different numbers of STRs from their parents. So 2 bands on their gel. Okay, so for one set of primers, okay, each people are going to have 2 PCR products. Okay, if they inherited different numbers of STRs from their parents. So if they got one set of STRs from mom, one set from dad, they're going to end up with 2 different bands on their electrophoresis gel when we run the electrophoresis gel. Sorry, inherited different numbers of STRs from each parent. And so, like I said, that's going to result in 2 bands on the gel when they run this through the electrophoresis. Because remember, these could be different lengths. Okay, and so let's look at an example of how this would work. So we've got this photo over here on the side, and this gentleman here was in prison for 17 years for a murder that he did not commit. And he was exonerated based on his DNA sample, based on STR. Okay, and so what you have in the chart here, you can see that these STR markers, so there was DNA on the victim, and they had STR markers of 17 and 19. Okay, and so Earl Washington was the gentleman that was freed. That's this guy here. Okay, he had 16 and 18, and those don't match, and they would have to match. Okay, for example, this means that he had 16 repeats of this STR, of these short tandem repeats. He had 16 of them on one chromosome and 18 on the other. Okay, so he got 16 from mom, 16 from dad. The victim, the DNA that was found on the victim, they had 17 and 19, and that immediately eliminates him. Okay, he cannot have done this. That is not his DNA. They would have had matching STRs. Okay, and you see this gentleman on the bottom here, this is who was finally convicted of it. Okay, and so they found the first STR marker, he matched. And then they would need to do it multiple times, okay, because that could have been a coincidence. But you'll notice every single one of these, they match. And so he was eventually convicted of the crime, and Earl Washington was released based on using this PCR to amplify this DNA and then comparing that amplified DNA, looking at these short tandem repeats, okay, and comparing that on the gel electrophoresis. We can also do what is called DNA analysis. And so with DNA analysis, you're basically looking for a specific sequence, looking for a specific order, and seeing if that particular person has that sequence. For example, you could look for a particular disease or disorder, like the gene that codes for Huntington's disease. Okay, and so looking for the gene that codes for Huntington's disorder, you're looking for that particular sequence and seeing if the person has it. This one would bring up another ethical concern, okay, as to whether people should be required. For instance, Huntington's disease is a lethal disease, and symptoms don't usually show up that you have Huntington's until about mid-30s, so about 35 or so. And once those symptoms show up, that person is on a relatively rapid decline. And like I said, this is a lethal disorder. And so there are some people that say, with Huntington's, it's in the DNA, so you pass that on to your children. And there's a large majority of people, most people reproduce before they ever see the symptoms of Huntington's. And so there's the question of, should they be required to see if they have this gene, especially if it already runs in their family? Should they be required to see if they have this gene before they have children? Okay? But then you also run into the other side of the coin of, can you require someone to do that? Okay? So again, just keeping in mind where you would draw the line on some of these debates for things. And so a few other techniques kind of just to make you aware of them, that they exist, would be things like xenotransplantation. Okay. This is from species to species. So for example, taking a pig val and putting it in a human heart. Okay, so using animal parts for a human recipient. Okay, so that would be your xenotransplantation. Things like injecting cells, injecting things into, for instance, example, injecting human brain cells into monkey brains to do research on brain disease. Is that okay? Manipulating embryos, okay, so of animals. So we've got an example of injecting sheep fetuses with human stem cells. So adding human stem cells, they can incorporate into the sheep fetus. So the sheep now has a liver that's made mostly of human tissue. And so now that liver could be harvested from the sheep. And so now you have, but now that sheep has no liver. Certain kinds of manipulate plants. Example of what's called BT crops. Okay? BT crops contain genes from a bacterium, okay? Bacillus thuringiensis. This is a tough one even for me to say. Okay, and that particular bacterium produces proteins that are toxic to pest insects. But is that okay for people to be eating these genetically modified foods? Okay, or another one called Roundup Ready crops. Okay, and Roundup Ready crops, these contain genes in them that protect these plants from herbicides, from that weed killer Roundup. Okay, but then that goes back to, again, this is a major food source for people, and should we be